way. And so this is where we're at, and we are, I have your exams. I'll hand those back at the end of class, and we'll talk about some more in class. And I will, we did our in-class assignment on Tuesday, and I already mentioned if you want yours, you can get it back. I got those scored, and some people emailed pics. I got those scored and sent back to you. And then we are moving ahead. And so when I hand your exam back to you, there'll be a circled score on the last page. That's how many you got correct out of 100. When I grade exams for all my classes, I do this. I make the grades, and then I make an anonymous distribution of the scores. Here's an example of that from Intro Bio. And each student is represented by an asterisk. So on that exam, there were 80 points, and so the highest student got a 74. And so there's one student. Here are two students got 64. Here are three that got 57. And so you can see that when I post on Brightspace the recorded today, I'll also post the distribution of scores for our first exam. And it'll look like this, fewer students. And you'll be able to see, oh, if you got a 60 points on the exam, you got a B minus. And the way I grade is it's not a 90, 80, 70, 60 cutoff, and I never do anything like a curve. I grade more like I think an English professor would do, where they, examine the, work, and then they say, oh, that's an A work, that's a B work. Okay, and so you'll see that, and two things are helpful. You'll see this was a hard exam, right? Students didn't do as well as they wanted. That's true for the class. You'll see that, so you'll get some comfort from that. You'll also see where you stand relative to other students in the class. That's the basis for your grade, like that. And you'll see, that, you could get better. If you got 10 more, you would have got, a B plus, like that, right? And so the grades shift around a little bit for each exam, but it's what it looks like. And so you'll get a score today. You'll be able to have your exam, get it back, and you'll see your letter grade when you get this link. I made an answer key video, and I'll also have that link there. And so you want to go through the exam, and that's a big part. And I'm actually next week going to have an assignment where each student goes through the exam, and so be aware of that. And so hang on to your exam, because we're going to go back through it so it can be a learning experience. And two types of learning experiences. One is to understand the material more. Three types maybe. Two is to understand the learning, because I consider this a lot about how you learn, trying to encourage you to strive to learn how you learn. And then three is also learn the format for these exams, because the second exam or the final exam, we're going to have that same style, and so this can be helpful for our upcoming exams, how you prepare and take the exam. Okay, and so that piece, so you'll know what your letter grade is, and that'll give you a really good handle on how you're doing in the course. Now, the next thing is I've taught this class every fall for 22 years, right? So I've taught this a lot, and it's always the same. No matter how many assignments I put at the front end, the first exam is always surprising, because students are like, whoa, like that. And so this is true. And over the years I've taught this, students end up with a final grade that's typical of their science final grades. It's a hard science class, like your hard science classes in chemistry and biology, but it's not one that's going to crush you differently than some other classes you take. It's just one of your hard classes. Why? Because it's introducing such a huge chunk of biology. Yesterday we were talking about sponges. We're talking about all animals on Earth and how they're all put together the same way. We look so different than a sponge. We even look different than a fish, right? So different than a fish. But yet, down at this molecular, cellular level, the stories are common because of our common ancestral origin. Charles Darwin wrote The Origin of the Species, almost 200 years ago, and in it he wrote many times. He said he knew embryogenesis was a big part of the story of evolution on Earth, but he just didn't appreciate how. He had tremendous insight, right on target. there was a piece missing. And a piece missing is that the eye does not evolve. The formation of the eye is what's evolving. Because each generation, a whole new eye gets put together through the inherited information in the DNA. And so that plan is what's evolving. And so it just puts a whole different spin on evolution. And now that three-legged table of biology, which is all about evolution, that three-legged table is super solid for the first time in history because of these discoveries. So, you know, we're trying to introduce a lot of fundamental stuff, right? And doing my best to do that while also encouraging you to learn how to learn, because I hope it's stuff that you can keep for the rest of your life, like that, you know, because there are things you'll keep for the rest of your life, right? You know, when you're in a nursing home, but somebody can say, oh, he spilled some water, and you'll be like, oh, H2O, like that, right? And so hopefully this will stick too, like that. Because when you go on a nature walk too, you'll be like, hey, look at that, look at that. We're really similar in a lot of ways. So you'll see your grade, and grades will go up. You know, your scores will go up, but it's your grade. And that, you know, we've had some assignments, so those strengthen your grade. But you know, this gives you an indication of where you stand, and you know, you'll be wanting to go up, and just about everybody will. You know, and go up significantly like that by doing better on the exam, and also the other assignments, and also this term paper that we're going to deal with. So, so we're dealing with chapter 2 this week. Another thing that happens in this course is I see biology as being way too many examples, way too many details. And so anytime I read biology, I'm saying to myself, what's the take-home message here? Always. Because I'm starting to stack up a sort of a framework. And it's always a physical thing. It's always a physical thing. And I'm starting to build a framework, and it's like, I think it's like putting up a Christmas tree, and then I start decorating it with the different facts. You know, the different ornaments are very appealing, and that's what you pay attention to, but you gotta hang them on something. And so you gotta read it, but my gosh, we're gonna talk about what we need to know, and that second chapter of our book could be much better organized by framing it into these 3 stories, because the author is literally telling these 3 related, somewhat overlapping stories about early fruit fly embryogenesis. So we're doing that as a sort of model, and that's a big reason why we read the book, because it can help you understand the stories. You can also see how the story could be kind of spread out in a different way, and you could go through it with a framework from class and be like, gosh, I see how this is starting to put together and be a physical memory somewhere in my brain. So that's that. Bring questions to the class always. Then go over exam 1, you'll see your grade, you'll see the answer key video. Save your exam, because we're going to loop back on that and do an assignment, you know, figuring out some of the stuff that we can do better on, and we'll do that next week. Because the thing you should do, and it's not, won't take very long, is to work on fruit flies, but also work toward this term paper that everybody's going to write, and we're going to focus, each student is going to pick article, and it's going to be a research article from that journal development. And so that's in your syllabus, and just bring like the first page of it to class on Tuesday, so you make that step, and then we can look through them, and I can just say, you know, Zach, you have a paper, and I can pick it up, and I can just read the title, and after I can start giving everybody sort of a feeling for how we're going to tackle it. What our goal is for you to connect it to our course. Like that. So it'll be about orthologs. So you can start out your paper and write a paragraph about orthologs that you already know about to connect it, okay? So that's that. So again, on Tuesday, we did this foreshadowing into chapter 7. So we're gonna do fruit flies, then plants, then this chapter 7, which is about cells, and it's animal cells in development. And one of the stories that we'll learn quite a bit more about there is these proteins that are on the surface of every one of every animal cells, and they include proteins that you might have never learned about before that are called cell adhesion molecules, and they hold hands with other cells. They also make contact with some other material we'll learn more about in chapter 7, and they're just hugely central in the story of animal development. So we'll get to more of those, but just to show you this, sponges have those. Sponges can self-assemble, reassemble if you pull them apart, just by those hand-holding molecules, and how there can be different ones in different cell types. And they are the most distantly related animals to us. So what they have that we have is just stunning to see. But that's how animals first started getting built, and we still use some of the same principles. That's the message. So we'll get to that chapter 7 pretty soon, and we'll learn a lot more about those topics. And students tend to really like the cell biology of development because it's so accessible, because it's like, oh, you mean hands really literally holding each other? So we did that in fruit flies. And our story of how we make compartments of different populations of cells is different. We don't use the same story because the fruit flies and us are really distant related. We have a lot of orthologs. Those are tools like Wnt and sonic hedgehog. We have a lot of the same orthologs, so those are worth learning, but we use them in different contexts. Okay, and so maybe I'll show this first. And so here's just one of the students' work from Tuesday, and I wanted to show this because, you know, anytime you're doing stuff with your hands, you're kind of like distracted, right? Because we don't do enough of that in class. You did a lot of that in kindergarten and first grade, but we kind of left that behind, even though it's such a great learning tool. So just to loop back through that, the posterior cells are shown here. Here's the compartment boundary that's in the sort of the visible line. It's in the middle of every segment of every arthropod, from, you know, lobsters to ticks, like that. And so that segment is organized by an invisible line in the middle that's called the compartment boundary, that's separating a population of cells in the anterior and posterior. And the posterior cells are in the posterior because they're all holding hands with cell adhesion molecules that are similar. And so that's why you use macaroni, just to bring that new concept up and have something for you to remember. Macaroni is cell adhesion molecules, and there are lots of different kinds of macaroni. So this student uses elbow macaroni for the cell adhesion molecules. And, you know, we were building this as we went, so a lot of students didn't really get a chance to sort of depict that there are multiple posterior cells, and they all have the elbow macaroni on their surface. And where they're touching, they're making that contact, and there are non-covalent bonds between those elbow macaroni molecules that are holding on and keeping them together, like holding hands. So that's one of the big visuals and the big takeaways, is that posterior cell adhesion molecules are holding hands. So that's one of the features of the identity of posterior cells. So let's just go back to this, because we've been talking about examples of how cells become different and what it means for them to be that type of cell. So what does it mean to be a muscle cell? And the answer is, well, you're making muscle genes, and you make the muscle fiber proteins. So you're a muscle cell. It's like almost a circular definition. What's a muscle cell? It's expressing muscle genes. What are muscle genes? They're expressed by muscle cells. People tend to call those genes that get activated during differentiation of the different tissues, they call those tissue-specific genes. That's just the term they use. So there are some muscle-specific genes, some brain-specific genes, some pancreas-specific genes. There are lots of genes that overlap, but that's a feature of our bodies, is we have many different types of tissues, and they all have all the genes, but they're expressing some different ones that are tissue-specific. So we have muscles. Then we talked about stem cells, and we talked about how stem cells are special cells, and we could actually take any cell and turn it into a stem cell. And the way we would do it is by somehow activating a set of genes that cause the cell to become a stem cell. The cell has all the genes, it's just not using them. So can we force it to use stem cell genes? And it's just a little different term that people use, but it's the same concept. Those Yamanaka factors are stem cell identity genes. They're the genes that are comparable for stem cells as MyoD and myogenin are for muscle. MyoD and myogenin are some of the transcription factors that activate muscle-specific genes. The 4 Yamanaka factors are transcription factors that activate stem cell-specific genes. And so you can see, you know, if you just dial back like 15 years, that Yamanaka had that insight. You know, he saw that fact in life on Earth, and he thought, well, stem cells must be expressing specific genes for them to be stem cells. And he worked on defining which those are, okay? So that's an example. Then we give some brief examples. So we just said briefly that belt that we wanted to make on the frog, You know, we talked about how it gets induced to become a precursor for mesoderm. That mesodermal belt, they're all expressing brachyury. And brachyury is a transcription factor that's comparable to myodine, myogenin, or the Yamanaka factors. It's important for turning on genes that are mesoderm. Now, the mesoderms are it's temporary. They're temporarily mesoderm. They're gonna head on have fates to be muscles and tendons and ligaments and bones. But when they're mesoderm, they're being certain cell type and expressing those kind of genes. So our new example with the assignment fits into this. And the book calls those segment polarity genes. The newer, kind of more popular term would be compartment genes. Really, compartment boundary is an invisible line. It's really significant, but it's created just by having compartment cells, posterior cells and anterior cells. And so there's a set of genes that would be specific to the posterior. And so on the assignment, you can see, and you know, everybody did a great job, but this student did a good job making a little table of that list we were working on. Because these are going to be genes that would be needed to be expressed in the posterior, and they're the equivalent of those other stories. You know, what genes need to get expressed in the muscle, what needs to get expressed in the mesoderm, what needs to get expressed in stem cells, what needs to be expressed. And of course, this is a limited list, but it's a sensible list. And so let's just talk about that. You know, so yeah, well, it's going to have to receive the Wnt signal. So it's got to have the receptor and the signal transduction pathway, and then that beta-catenin. And those are all proteins, so those genes have to be activated. And beta-catenin is one of those. So beta-catenin is a transcription factor that in this setting is playing a role comparable to myogenin, myodine in muscles, or brachyury in mesoderm, or the Yamanaka factors in stem cells. It's going to activate a bunch of these genes, so we become posterior cells. And one of the genes, just to include it, is engrailed. And engrailed is a transcription factor. So one of the things that these do is they'll activate another transcription factor so they get help turning on the necessary genes. We saw that at the start of our course where MyoD is going to activate another transcription factor, myogenin, just to get help turning on the genes. So there are more transcription factors that get turned on the posterior, probably, but this is a good principle, is, you know, a beta-catenin is going to get help turning these into posterior cells. How? Well, activate a transcription factor like engrailed, and engrailed can team up and help you. Then, the next one, sonic hedgehog. The posterior cells need to shout to the anterior, be anterior. That's part of the plan. If being posterior is making somebody else anterior. And so they got to make that sonic hedgehog signal and release it. Something that's not on the list we didn't talk about is they aren't responsive to their own signal, right? They don't have that patch receptor. They don't respond to the signal. They just release it. They're a source cell, and the target cell is the anterior cells, like that. So that's a good one because it's going to set up this feedback loop, because the anteriors are shouting at the posterior by making Wnt, and you get this signaling feedback loop. And then the next one is these cell adhesion molecules. Not just any, there are lots of different kinds of macaroni. This one is activating the elbow macaroni cell adhesion molecule gene. And that's a really important part of being posterior, because that keeps the team together. And the idea of the team is to create a difference, making a molecular, or in this case, cellular switch. So we start out with a fertilized egg, dividing, and the cells have to start to become different. They're all going to have all the genes. How are they going to become different? And we've talked a little bit about gene expression. This is about how the cells themselves can behave differently and help create a crisp switch, or a crisp line that we can use as the basis for all the rest of the development. So great. And then the last one was just to help you understand that this is going on for the entire development, because it's activating signals along the line that spread out further, and DPP is an example of that. Spread out further to help pattern that overall segment. In fact, where there's very low levels of DPP, the cells create a segment edge. The book calls the compartment boundary the parasegment. which is an old school, very fruit fly specific term, because that's actually the original design plan. The design plan is, let's have some repeated units along the length of the body. Let's do that by having a line in the middle of these repeats. And that middle is going to become the segment. It's going to become the segment by establishing edges. This is the farthest edge of this segment. That's where we don't have any DPP. This is the farthest edge of the other end of the segment. That's where we don't have any DPP. So it's pretty, it's physical, it's memorable, and that's a key thing, because on the second exam, I might just focus in on that and say, what's a list of genes that the posterior, cells need to express? I could have you draw part of this or have fill in blank. That's a really nice list. And of course, there are more genes, but it's a nice list because it ties into the other things we're studying and also brings in this new one of cell adhesion molecules. Oh, did I not? Yeah, did I zoom in on this? I tried to. Yeah, so see there, I should have shown it, but a nice little table, and of course, the name, and then what's the role of each of those is a good thing, for you to let sink in, because it fits into this specific example and the broader idea of cells becoming different and becoming specific over time. Then the student also has some of these drawings of the genes, because it's all about these genes. They got all the genes and they're turning some on, and they're using transcription factors like beta-catenin and engrail, and some others too that we didn't learn about, to turn the genes on. So it really goes back to, I could have started the course with a clay model video of this and showing, some clay model of some gene here. But muscles is just way more familiar. So let's start with something that you can kind of tie into something and then stretch into something that's totally weird and stupid, like a fruit fly. But that's actually fundamental in the story of life on Earth. And then again, the cell adhesion molecules, those things, people don't really typically talk about orthologs when it gets stretched all the way to sponges. They use a more general term, homolog, and they mean they're related. But we could call them orthologs, because that's really what's going on, is some simple animal like sponges evolved a plant, have its cells get organized based on some hand-holding proteins on cell surfaces. And every animal on Earth is just taking advantage of that and run with it. We have hundreds of different cell adhesion molecules in our body, and they're really famous. Yep. So just to go back over that, and again, by tying into the sponges, we're like, yeah, we're talking about life on Earth, all animals that have ever existed, for sure. Here's the anterior. There's a comparable story going on the anterior. We could make that list. We just made a little partial list, right? One of the lists is, we said, well, it's got to make wind. That's got to be part of the anterior story. Got to make some cell adhesion molecules. They got to be different. So let's use some shell macaroni. And we just didn't fill that out as much, just because it's plenty, it's plenty. But really appreciate that the anterior population cells are expressing comparable but different examples of an anterior. So we got to have the receptor for sonic hedgehog, and it's a protein, have to be called patch. But that's more confusing details to go over there and say, learn as much about the anterior as the posterior. When we talk about it, let's really focus on the posterior and then be able to say, yeah, the anterior is doing comparable stuff, but different genes, different cell adhesion molecules, different receptor, different transcription factors. CI is a big transcription factor in the anterior that's comparable to beta-catenin in the posterior. And CI is playing a role like MyoD does in starting to make muscles, or like the Yamanaka factors are doing in starting to make stem cells. So, CI, that's just as important as anything, and so forth, like that, okay? And I just put a little vial out there of fruit flies, because fruit flies, S-U-C-K. I'm just kidding, I'm kidding. I studied them in grad school. But most people are just like, oh my God, you study fruit flies? Why don't you go outside and go for a bike ride? This isn't from our book, but it's from another book, and it has a really nice picture of this, and it's just showing that same thing. It's just showing one anterior cell, and there are all of them, one posterior cell. What it's emphasizing is initially, and our book shows this too, initially it happens that this gets set up when the segment is only 4 cells wide. So that's very early where the cells start to decide, we gotta set up a plan, and this is an anterior to posterior plan. And the plan is, let's slice this thing up like we're slicing up a loaf of bread, and we'll make slices of segments and organize each segment. And we'll go on to have some segments that are different, like this segment's gonna have some wings, this segment's gonna have some legs. Yeah? Okay? It's all based on this, so we learned about this in a weight drawing assignment, and we were plugging that learning in. This is in chapter 2, that's the sonic hedgehog signaling pathway. We don't need to know that in such detail. Wnt is our favorite one, but just appreciate that, you know, sonic hedgehog binds to a receptor on the cell surface. A signal transduction pathway is going to affect a transcription factor like CI, and CI is going to regulate target genes. Okay, great. So that's what's showing there. And here's our book showing this, and it's a kind of a confusing image, but it's showing that idea that this happens very early. The time is about 2 hours after fertilization, so it happens very early and very fast. And you can see here, the book is doing a good job showing you that these are the segment boundaries. So let's just pick one segment, right? So there's the segment boundaries. And this is like what you'll see in the future lobster tail, the individual segments along the lobster body, it's segmented, or any insect in a caterpillar, you see the multiple segments really easily. That's one of them. And it's getting set up here. The original is this compartment boundary, and we love to call it the compartment boundary, and there it is. And you can see initially, they're actually, it turns out, kind of surprising, 3 cells that are anterior cells and 1 cell that's posterior in each of these segments. It's just repeated along the anterior posterior length of the animal. And it's just showing you what are some of the genes that are expressed in each segment. It has a typo here, that's fine, but it's showing hedgehog, and hedgehog is expressed in the posterior of each segment, and it's coming to the anterior. So it's showing what we learned in our assignment, just so you can see that. And so that's our first story, and we're done with that story. And I again want to say that our bodies are set up like that too. We don't have obvious segments like an arthropod does, but we have a spinal column that has very clear slices of vertebrae from anterior to posterior. And they're different as you go from head to tail, right? You have your cervical, thoracic, lumbar, sacral vertebrae. So it has that plan. These somites are also set up to be compartments, and boundaries are set up, but in a very different molecular way. They include things like cell adhesion molecules, but using different kind of strategies. But the same concept is seen over and over again in animal development. If, let's make teams, you know? You got a bunch of people, let's put them into 2 teams and move on, you know? And we can have plans with the 2 teams having a line between them, and each team can go on a little different plan, like that. Here's our central nervous system. Both these images are from chapter 5, parts we didn't cover, but the book, covered. Here's our brain. Everybody knows the brain goes hindbrain, midbrain, forebrain, midbrain, hindbrain, spinal cord. So those are chunky compartments. This is focusing on our hindbrain because our hindbrain is famously set up into 7 pieces from anterior to posterior. They're called rhombomeres. We didn't cover it, but I just want to show you this because even though we didn't cover it, our bodies and every animal has a lot of these plans to make compartments. They might be rhombomeres in our hindbrain, they might be somites in our mesoderm. There's a lot of that's going on. So we have a couple of things that are happening in that fruit fly development story. One is the introduction of the cell adhesion molecules. Another is the idea of let's organize this into populations and build cellular populations that are distinct early in development. 2 hours after fertilization in a fruit fly, we set up teams and we say, let's keep these teams, let's go. Yep. So that's different because it's different plans, but the fundamental, deepest concept is seen over and over again. So now here's the second story. And we can just look and see the second story is going to be about setting up the overall anterior or posterior axis. And then the third story is going to be set up the dorsal ventral axis. We do it very differently in our bodies because of the last common ancestor between us and fruit flies being such a simple little worm, really simple little worm, that it had an anterior and posterior, but it hadn't really developed it very much. It's clear that it was starting to have like a head end, you know, with more of a mouth at that end and more move that way, but still very simple. So there's lots of differences between how we set up our anterior posterior axis and how a fruit fly does. Same with dorsal ventral. You'll see there are a couple of features that are similar, and here's one. The end of this second story is the fruit fly is going to be expressing these different Hox genes in the anterior posterior axis. And that's the same in us. The interpretation is those are orthologs. The last common ancestor between us and fruit flies 600 million years ago must have already had this Hox gene plan for establishing in that ancestor a really simple front end to back end work. But most of the other stuff had been established, but that's just an old one. And what does it mean? It means that's a great plan. Let's make a little animal that's got a front end and a rear end, and it's going to kind of move that way and be looking for food, you know, like that. Yep. So we'll get to that. But how we get to Hox gene expression in us and fruit flies, how we get there is completely different. There are some genes that we'll see that are used in our bodies too, but used for different purposes. And that's similar too in the dorsal ventral axis. We do that too, but in a different way. The last common ancestor must not have had much of a difference. And worms don't. You know the difference between their tummy and their back and a worm, earthworm, stuff like that, really simple worm, is really not much difference, right? So difference, and so not much commonality in how we do that. Here's a handout I have, and I will send that, I'll post it with today's thing, but I printed it out because it's not one of these handouts where you might want to look at it as we're talking. And so you can just hand these around, and it's got these two images on it. The first is my attempt to make sort of a minimal list of genes that fit into the story. Because I mentioned in chapter 2, the author covers 50, 60, 70 genes, and they don't all matter. So, you know, here's an initial minimum that matter. And I told this second story first, but here you can see we learned our story about the compartment boundaries, about sonic hedgehog, about wind, and grail, CI, decaplegic. So those are some examples of the most hardcore, significant genes, you know, that you'd be like, I'd really be hoping you'd learn them, and maybe a couple more. And so you can see the next story we're going to teach is here, and it's a list that goes in order of expression. Then down here, we'll talk to that. That'll be dorsal ventral. We won't get to that today. But this next story is going to go like from the beginning, early, and later. It's a timeline. And that last one, Antennapedia, is a Hox gene. And then the next thing to say as way of introduction is each one of these is a transcription factor. And so each one of those is going to play a role like that MyoD, then myogen, then the next one. They're going to turn each other on, and what they're doing is initially the early embryo is at one end and another end, and they are quite quickly slicing it up into pieces. The Hox genes are going to set up the differences between each segment. So one of the segments will have wings. Why? Because certain Hox genes are expressed there. So that's our goal in establishing anterior posterior in this story is get to turn on the Hox genes in a direction from head to tail. And the Hox genes will be like, you should be head, you should be thorax, you should be abdomen. And it comes out like the first one comes out, and the first one just kind of starts splitting the embryo up into a broad sort of one end to the other end. Then the next slice it into sort of four pieces. One way to think about this is think about this. Imagine that you sliced a loaf of bread this way. Instead of slicing it the normal way end to end, you slice the bread like this. Here's a loaf of bread. I'm going to cut it in half. I'm gonna cut it in quarters. I'm gonna cut it in eighths. That's what's happening, and these transcription factors are turning each other on and getting more detailed. And they're gonna be activating some and suppressing others to try to create these molecular switches to make crisp slices of bread. Yep, so we gotta talk through that, and in talking through that, this is the figure from the book, and the figure from the book goes from early to later. And this bottom one is the Hox genes. So we go 1, 2, 3, 4. I just would draw this on the side because the authors, I think, have sort of clumsily combined our first and second stories. Because these genes here that's our first story. And then this is our second story. So this second story we're talking about now, that's the one we'd say about the Hox genes. So we're going to set up the identity of each segment. This one, here the author is referring to as segmentation genes. A better way to draw this would have been to draw a pyramid, where this is the first one, and that's the second one, and that's the third one, and the next ones are here, side by side. Yes, we need segments. So let's use the segment polarity genes to create those compartments. And that's where they fit. And then we need the segments to be different. So let's have some Hox genes expressed in different ones along the anterior posterior axis. And this would be a timeline from fertilization to about 3 hours after fertilization. It happens really fast that you get this cascade. All the cells have all the genes, they're just not using them all. Okay? So that's the flow of what our goal is at a molecular level. And then, if we go back to this list, this would just be an example of the top of the pyramid, the next step down, the next step down, and then these would be at the bottom of the pyramid, side by side. Yes, we want some segments. Yes, we want some segments to be different. So that's the flow. And, it's interesting to see, because the authors, talk to artists, and artists create these figures, and you're like, dang it, that could be better. And, you're new to this, but I'm just like, wow, shoot, I would like to teach this, but it's hard. So it's hard to navigate through books sometimes because it's just true that they don't have a really great explanation always to help you, the student. Yep.
So is in the drawing on the left, segment polarity is next to Hox, they're both, one section at the bottom is segment polarity, the other section is, okay.
Yeah, right. And this is like an abstract figure that's showing the time, but not showing the body. It's not showing that these are in part of the body, they're in another part of the body. They're both parts of the whole body's anterior to posterior. This is saying, let's set up some segments. That is saying, let's make the segments different. Like that. Memorable image, I think. So to get there, we actually have to talk about the fruit fly female and what they're doing. So that's on the handout too, not in your book. Our book doesn't show this, but anytime you can do a physical explanation, I just love it, right? Because we're gonna go to the fruit fly's body before dad is there. Because the fruit fly is gonna make a great big egg and gonna load in a bunch of maternal factors. Our story starts there with that first one being a big time maternal factor. And it's gonna start turning on in the embryo zygotic gene expression. Like that. So we're gonna get a cascade of gene expression. Okay? And so that's great. Our story's gonna start, because it's so well characterized, it's actually gonna start in the ovary, and it's gonna start at the anterior tip of the ovary, where there are a small number of stem cells producing all of the eggs. So they're dividing, and they're producing precursor to the egg cells that divide through meiosis to make an egg cell. And so that's our very first part of the story, because it will make sense. So I'll just draw over here to give you some landmarks. So like here's a female ovary, and it has an anterior posterior end, and it's just a little ovary. And at the anterior end, there are some stem cells. And then at the posterior end is where ovulation takes place. And ovulation is when the mature egg cell is released from the ovary into the female's reproductive tract. It's an oval-shaped egg, and it's then when fertilization will take place. So everything we talk about here is inside an organ where fertilization cannot take place, and where the egg is maturing and getting all of the maternal factors and the yolk and the building blocks put into it. So it's a mature egg that can be ovulated, can be released into the female's reproductive tract where the sperm can fertilize it. A fruit fly female can lay 100 eggs like that. They lay eggs like that. They're just laying eggs like a machine. When they find rotting fruit, they're like, aha. And some of those eggs are stored, but they're making eggs really fast. A female fruit fly only lives a couple of weeks and only like a week when they are reproductively mature, and they'll produce 400, 500 eggs. So that's the next thing, is that this ovary is going to be like a factory for egg production. Okay? And so we're going to have stem cells up there. There's going to be a few stem cells dividing, because we know stem cells can divide like this. They're self-renewing. And these are dividing asymmetrically to make precursor cells. And each precursor cell is going to become an egg. Before it does that, the precursor cells are going to each divide 4 times by mitosis. So even before they go through meiosis and make an egg, they're going to go through 4 mitoses and make some cells we'll talk about. And so there are a small number of stem cells here, and each one is at the tip of a factory of egg cell production. So that's just one stem cell, and this is showing that it divided and made a precursor cell, and then that's dividing to make 16 cells, and then that's the older one, and the next older one, and the next older one, and the next older one. And that string is just like a timeline of factory work. Okay, does that make sense? That's the oldest one, and it's ready to go. That one's not quite as old, not quite as old. And so, 1, 2, 3, 4, 5, 6, we're showing the stem cell is divided 6 times. And it just keeps doing that, and then you're ovulating these eggs. And one of those strings of egg production factories is called an ovariole. It's a fruit fly. It's not the world's most important term, but to help kind of frame what's going on, we'll talk about that. And so up here are these stem cells, and they're just churning along. These stem cells have a special term. They're called germ-line stem cells. And germ-line is a general term for cells in animals, animal cells, that can make eggs or sperm. That's the only cells in the body that can be sent on to the future. In a sense, our germline cells we have in our body so far in history have been immortal. It's the germline cells progeny that get passed on, the sperm and egg. So a sperm and egg unite, form a body, and in the body there's a set of special cells called the germline that can make the next generation. The rest of the cells in the body, including cells in the ovary and testes that are not making sperm and egg, those cells have a different name, and they're called somatic cells. And so our brains, our liver, our pancreas, those are all called somatic cells. They can't make sperm or egg. In our testes and ovaries, there are some somatic cells that are helping, and then a small number of stem cells are these germline stem cells. And that germline is going on continuously. Our somatic cells are mortal. We're gonna die. We could maybe send on some cells to a future generation, children and grandchildren, great-grandchildren. That's what's been happening so far. Those are germline cells that get passed down. Scientists will say this cute little quote. They'll say, a hen, chicken, is just an egg's way of making another egg. Our bodies are just suitcases carrying around germline cells, literally. So, distinction here is a good place to create it. The germline stem cells. And they're just trucking along making eggs in this case. Eggs, eggs, eggs. And there are several ovarials. There's one ovarial for each of these germline stem cells. And they're dividing asymmetrically, and they're just creating as many eggs as they can. So you can see in this figure we have, there's an ovarial, and colored in there. So there must be a stem cell up there, divide, and then you can see what it's producing is this yellow egg cell that's in a bag, and it's not showing all the cells here, but in the bag are 16 cells. Each of those oval circle bags has an egg cell and 16 cells. So we're going to spend a lot of time drawing an egg maturing, and this again is anterior and posterior. So when I draw it, I'll tend to draw it sideways like this, and that'll be an egg cell. Okay? And the egg cell is the yellow thing, and it's going to be surrounded by two types of cells. The 15 other cells in that bag are called 15 nurse cells. And you can see nurse cells, they must be germline cells. But they're called nurse cells for a good reason. They don't make an egg. The nurse cells help that egg get ready. So each one of the nurse cells actually has a tunnel, a little hole in it, connecting it to the egg, and they're sending, the nurse cells are helping make proteins and mRNA and building blocks, and they're just bringing them in on a train, because this is all happening fast. We've got to make a big egg with nutrients, building blocks, amino acids and so forth, but also maternal factors. And that first one on our list is a maternal factor called bicoid, and bicoid is a protein, and bicoid gets the gene gets transcribed in the nurse cells. A bunch of genes do, and that's actually where it gets made, is in those nurse cells. So we gotta learn a little bit about the nurse cells and how they're connected and what they make. And our one example of all the things they make is this bicoid. And interestingly, the nurse cells transcribe it into mRNA, so there's some bicoid mRNA, and that is what they deliver, in this case, to the egg cell. And we saw that a little bit in the frog, that the egg cell is going to mature and be ready to go, but waiting. And one way it waits is it makes bicoid mRNA, and it just puts that in the egg. And after fertilization, the bicoid protein gets translated in the fertilized egg. So it's a sweet example. If you said, let's just pick one example, one overwhelm people, because they're making a lot. They're huge cells. I mentioned that stem cell divides 4 times to make 16 cells. In those cells, the nurse cells just keep copying their DNA, because they are just going to be factories. And they want a lot of the bicoid gene to make a lot of bicoid mRNA to deliver it. And they want a lot of the other genes, because any maternal factor they're making, they want to deliver a lot. And they want to deliver a lot of nutrients and stuff. So they're big cells with lots of extra DNA, lots of extra chromosomes, and they're just cranking. Do our bodies do this? No. One reason is our egg cells are little, because we're mammals, right? None of these maternal factors, none of these nutrients get packed in. So this is a very much a non-mammalian story and non-vertebrate story. Yep.
When the stem cell divides to make the 16 cells, what makes the one cell the egg cell? Right?
Yeah, so let's draw that out, and actually, I'll show you here's an image I could draw out. So here is the precursor cell. It has a name we don't need to learn, but you saw us, right? Stem cells divide, precursor, stem cell divide, precursor. So there's the precursor cell, and this is just showing visually what it's like when it divides. And so that first one is going to be labeled, that's going to be the egg, and it's going through 4 mitotic divisions as its products. So we end up with a total of 16 cells. So one cell divides twice, you have 2 cells make 4 cells make 16, something like that, right? Okay, that's the basic idea, the concept. And these are all the nurse cells, and you can see they've numbered them 1 through 15. They've got 16 on here, which is flawed, but it should be 1 through 15 because the total is 16. And you can see how they all stayed connected. They've got a little tunnel there. Okay, and we'll just name that tunnel since we're on that topic. Each one is connected ultimately to the nurse cell, the egg cell sometimes. And so this cell could make some bicoid mRNA, and it would go through this cell and this cell and this cell. So it's going to get delivered. And the way it gets delivered is the cells are connected with a canal between them. So their cytoplasm, they're physically connected, and that has a special organ called the ring canal. Do we have anything like that? No, our cells are not connected like this. And through the ring canal are components of the cytoskeleton, and you can draw it like a train track because that's what it is. Those are microtubules. And microtubules are part of the cytoskeleton. We'll learn more about microtubules in chapter 7. Right now, it's part of the cytoskeleton you learned about in intro bio. And the microtubules' job is they deliver cargo. So it's great to always think of microtubules as train tracks. They grab cargo and little molecular motors drag the cargo along the path. And so that line I've drawn there is literally a train track running from the cell number 14 to the egg cell, bringing stuff from the nurse cells. And so it's it's got a special name, I guess I'll name it. This whole system that runs through it, the ring canal and all the train tracks all together are called the fusome. And it's fascinating because it's just part of this big factory story, where you're like, we gotta make a big egg cell, let's go. And so, right, you can see it like somehow the germline stem cell divide, and the precursor's like, I'm gonna divide a couple of times because I'm gonna need some help making this egg. And through process of evolution through natural selection, you're like, yeah, let's divide a few times and get some helper cells going. So the fusome is just literally that, and we can just draw on it right now. What's getting dragged on it is there'll be a little mRNA that's bicoid, and somehow that bicoid mRNA is identifiable by these motors, and they're just going to drag it and deliver it into the egg cell cytoplasm, and it'll be ready. Another thing we can draw right now is this, and this is just a real simple image, but it's a really important image. It turns out that figure showed how they're all connected. They're actually all connected in this way, where there's the egg cell, and these are nurse cells, and I won't draw them all, but here's some nurse cells. And the nurse cells are all connected to each other through ring canals. I won't draw them all. And there are actually 4 ring canals that connect the nurse cells directly to the egg. Again, the nurse cells have ring canals, but there are 4 ring canals, and they're all next to each other at one end of this oval egg. And again, mom is not even getting ready to go out on a date with dad yet. But we can already say this is going to be the eventual anterior end of this embryo. So mom, by loading in maternal factors, asymmetrically, unevenly to the egg is going to say that's going to be anterior. And of course, the other end is going to be the posterior end. And how does that happen? And we can say bicoid is a big part of it. When the bicoid mRNA gets delivered into the egg cytoplasm, the bicoid mRNA gets anchored and stops floating around. So the bicoid mRNA is all going to be there. And after fertilization, so now, maybe just make a special little thing and say, look, here comes the sperm, and it's after ovulation and fertilization. After fertilization, when the bicoid protein starts getting made, it all starts getting made up here. And in this big egg, it starts to diffuse. And so we can make a graph like this with a concentration, and this is bicoid protein, evenly floating from the one end of the egg to the other. Again, this is one big cell. That's the cell membrane that the ring canals are in. The bicoid mRNA is all anchored, all stuck in one end of the egg. When the bicoid mRNA starts getting translated soon after fertilization, you get a lot of bicoid protein there and not so much back there. And bicoid's a transcription factor. It's going to turn on genes. It's going to be turning on genes at the end that's going to be the anterior. So just like that, mom sets up that anterior-posterior axis, future anterior-posterior axis. So our story that we're telling is all about this. It's about the plan of let's take that egg, and after it gets fertilized, let's start setting things up. So we can start slicing this up like it's a loaf of bread, and slicing it up in that surprising way of like slice it in half, slice it in quarters, slice it in eighths, and we're going to make 14 slices for the 14 segments. And we're going to say, let's start setting up, so that's why I said our macaroni story, it starts like 2 hours after fertilization. Because that's where we start having 4 cells wide, and they're like, we're going to be a segment, like that. Yep. Yeah.
It says bicoid mRNA gets, and then what's the last?
It's partying. Let's see. Bicoid mRNA gets anchored.
Anchored. Okay,
Yeah.
Thank you.
So it doesn't just float around, it gets stuck right after it gets over the egg cell. So it's coming in on those tracks, and they're like, you've made it to the egg. And then it gets stuck. It gets bound to another component of the cytoskeleton, and it stays there waiting. If the egg's never fertilized, that's where the bicoid mRNA is. It's just there waiting. Yep.
So does the bicoid come from the nurse cells?
Yep.
Goes into the egg cell?
Yep.
Okay.
Yeah. And with the ring canal, it's doing something that's unusual in animals, and that is animals don't typically share proteins or mRNA, RNA. Each cell has to make their own, typically. There are only a few examples where the one cell can share with another. And this is an example of that, where the cells are connected through a little tunnel, like it's like a little short drinking straw. And so that's cytoplasm, from cytoplasm to cytoplasm, the protein go through there. There are gates there. It's a really interesting place. It doesn't let everything through, and what it lets through is also getting pulled on these train tracks that run right through the little tunnel, like a train tunnel through a mountain. Huh? Yeah, yeah. Super physical story, accessible story, and we're decorating it, as the framework. Where is it happening? Are we talking about a human body? No. We're talking about a female fruit fly's ovaries, and in the ovarial node factory. Yeah? Yeah? Okay, great. Let us pause for a concept chat. So try to say hi to somebody new today. I'm sorry we didn't do this Tuesday when my friend Sarah was here, because she's a character. And but find somebody new, and if you can, and say hi, and try to have it be 2 people unless there's one group of 3, because it's uneven. And talk about some things, 3 things that you thought were important so far today.
Well, so it's that's the mRNA, and then once it fertilizes, it gets transcribed into protein.
Yeah.
And so there's a lot more protein with the mRNA, but it does, it's not anchored once it's a protein. So it's just a little more.
So it's translated, it gets,
Yeah.
got.
Let's see.
It's like
I
same
thought
structure.
that, what is it? The fertilization into like the box and segment polarity is really cool.
That
And how we like kind of illustrated it
there's
a little bit
16
more
cells
memorable
in each
for
one.
us. Just kind of see like how the segments kind of separated in that way. It was a lot of review at the beginning of kind of just like different genes that we'd already talked about, and then like kind of the difference between anterior and posterior in that sense.
And that's what,
I think that's the,
So-
yeah, at least everybody gets the same, right? Yeah, I think that's, like, everyone else likes to. I know.
I'm
It's
still
like
here, we're
the
still
part
talking about
focus,
flies.
yeah.
This segment, you see that, the coin is, what's the anterior axis.
Yeah.
So we're doing this for another minute. We're just going through this quickly. Talking about 3 highlights as part of this process of recalling and using your notes to help and stuff like that. Awesome.
I'm not sure. Is it a transcription factor? I don't know if the quite is. I know that it said in kind of is. So.
Yeah, that's what I was thinking, so might have just like help she's aware.
Yeah.
But this is kind of just the anterior axis.
Yeah.
yeah, so like the top of that, it might be the top of the
Yeah.
ovary, where the stem cells
Okay.
are.
You can check.
As the stem cell divides
So let us check in with Nathan. Hi, Nathan. What's one thing your group talked about?
So we went over both how the ring connect kind of transports the Bitcoin RNA from or mRNA, from the nerve cells into the actual egg cell.
Yep.
And then also the development of the stem cells in the ovary, because I was kind of confused about that.
Yep.
And so like sort of how that.
Yeah. Yeah, and this is a fruit fly story, and there would be other insects that this story is true in that have ring canals. But again, we don't have anything like ring canals. We don't make a big egg like that. And even vertebrates like fish and frogs and so forth, they don't have ring canals. They have some different mechanisms. Yeah. But this is a really good one because what they can do in fruit flies in telling a pretty simple story is they can tell the whole story. They can say, let's start with a stem cell and make an entire fruit fly. You know, because it's such a simple model organism. Chloe, do you have a question?
Yeah, it has to do with the stem cells. So I know they're self-renewing, but is there a point in like a woman's life where the stem cells are no longer being made, or like how does that work? Because I know like later on in life people can't
Yeah.
reproduce.
So there's a chapter in our book that we probably won't get to cover. It's maybe chapter 10 or 11. And it's about the topics of really like reproduction. So it goes through meiosis and production of egg and sperm and, in some detail. In mammals, there's a very interesting story. And it goes like this, just to fit it in. And again, we probably won't get to cover this topic, so it's not something that's going to be on an exam. So in mammals, remember you and I, when we were in our mom's tummy, we made an embryo that looked just like a chicken embryo. And we were also making, those are embryonic cells, we were also making what are called extra embryonic cells that make the tissues of the placenta and the amniotic sac and lots of tissues. And our moms made some of the cells and we made some of the cells. So in that sense, the placenta is chimeric, right? It's got cells from 2 different organisms mixed together to get the job done. When a woman gives birth, here's the baby, and about an hour later, here's the afterbirth. So that's the placenta and the amniotic sac and things, those tissues, extra amniotic tissues coming out, because they're not going to be part of her body. And I think it weighs about the same as a baby. It's a lot of cells. So that's the idea of embryo and extra embryo, okay? In us, right, you asked about a woman, right? In us, the germline stem cells arise extra embryonically. It's so weird because they're the only cells that matter. And they show up initially in mammals outside of the embryo's body, like that. And they're the germline cells. And they crawl into the embryo. And that's true for all vertebrates as well as us. And one of the parts of that is in vertebrates. And they're the most important cells. In vertebrates, weirdly, they crawl in different ways. In mammals, the germline stem cells get into the body by crawling in through the digestive tube, the archenteron, and are moving through that and get to the proper location and then leave it and find what's called the genital ridge. Those are the somatic cells that are going to be the testes and ovaries. In birds, they come in through the blood system. You know, so it's weird because they're so important, and you'd think they would stay this. Story's going to stay the same. And it's weird like that. Yep.
So where are they coming from?
Yeah. So there's, they come before this extracellular tissue has really differentiated into, say, placenta and amnia, just outside the body, but part of the embryo, not mom. These are extra embryonic cells that we made that then come into our body. They were out there going to be ready to make sort of the placenta and stuff, and then they come in. And so, yeah.
How do they know to come in rather than just?
Right, how do they know? So it turns out that scientists had a really hard time learning about them, and we have like 5 minutes, and so we'll just derail and we'll talk about stuff. Because a lot of these stories are like bedtime stories, because people literally call this the epic of the germline. Because another question is, how do they know how to crawl in? They don't know. They're going to be, right, it's going to be either a germline cell is going to be XY or XX, right? Male or female. Those cells coming in don't know what they're going to be in. Even though they're XY or XX, somatic cells tell them, I'm making an ovary, so you should make eggs. Or I'm making a testes, so you should make sperm. Yeah. So it's called the epic of the germline because it is like Lord of the Rings, where you're like, oh my God, look what's happening now. They have to go on an epic adventure. So it's only a few hundred cells that make the journey, and when they're making the journey, they're dividing. So they're increasing that number, some, right? Okay? And so people will say, scientists had a really hard time studying them. Here's how they got a way to study them, is they found a gene that's expressed in the germline, and they made a transgene that would make the GFP transcription unit and glow. So they could get the cells to glow, and then there's an instrument called a flow cytometer where they could gently separate all the cells in the embryo and actually pick out the glowing cells and study them. So that tool that we know about, a little bit about how to make a cell glow, was used to really start studying these. So here's some things. When those cells are coming in, they're dividing. Another thing they're doing is they're demethylating their DNA, because they've got to become an embryonic stem cell. And they were a differentiated cell. A sperm and egg are differentiated cells with DNA methylation to block expression of muscle genes and so forth. They got a whole genome, and as they're coming in, they're removing a lot of this and gonna recreate it. And it's just crazy. It's just a crazy trilogy, right? Like that, So then they arrive, and whether they arrive in a precursor to a testes or a ovary, the germinal cells, they famously stop dividing. And in a female vertebrate, let's limit it to humans. That's why people will often say, a girl is born with all the eggs she'll ever have, is because they stopped dividing when they arrived in a precursor ovary. And it turns out that that's somewhat false. It's not quite as dramatic as that, especially with some non-human species like that. So they're learning more. But the concept is still, the principle is that the cells all stop when they reach the gonad, and for the eggs, they're really stopped for good. They're gonna go through meiosis, but they're not gonna increase the number. On the other hand, for in testes, they've stopped, and they stay stopped until puberty. And then they keep dividing, start dividing again, because now they're gonna produce huge numbers of sperm for many years, right? Because there's lots of sperm, like that, you know? So in testes, those germline cells are dividing through mitosis to go the asymmetric division to produce lots and lots of sperm over the years. Yep. So that's, you know, the answer. And again, we won't get it there to it in our course almost for sure. But oh my gosh, all this is so fascinating and surprising, you know, because there's a lot of commonality. So you'd express something as important as germline cells, those must be the same. There must be a standard story for those. But yet, somehow there's a lot of evolutionary change that's occurred in the epic of the germline, of where the cells are and what they're up to. Yeah, really curious. Anyway, we have to stop. Oh, I have to end back your exams. I apologize. I was trying to give you some time for that. Okay, so you'll get a score. Hang on to these, remember, because we're going to do an assignment on them. And I will I'll get that stuff out to you. You'll see the answer key. We'll go through that. We'll spend some time on this. And I know, lots of you did do as well as you want, but I also saw everybody is solid. Everybody's on this course doing well, and you'll do over time. So there's Ellie and Naomi, and you pick out and you're set. If you want your macaroni assignments, you can pick them up. So Julia, Isla, I gave the one. Sorry, this is running around. Alex and Nick. And Chloe, and Nathan, and Jesse, and Maggie's not here, Jeff's not here, Ali is here, Zach is here, Brianna not here. All right, and so, just be gentle on yourself. I know some of you are like, darn, whoa. This is the kind of exam where students lose chunks of points. You're like, darn, I don't know. I didn't really look at that topic much, So, it's not surprising that we have, a big distribution of scores, which I'll share with you, and we'll move on and keep learning.